Hey, Mr. P here. In this video, we're going to talk specifically about membrane transport. So this is a follow-up video to the membrane structure video to kind of round out this topic of biology. In this video, we're going to talk specifically about how the membrane is structured, but ultimately how that structure translates to transport within and among cells. We're going to talk a little bit about the difference between passive and active transport. We're going to talk about osmosis. We're going to talk about bulk transport, and ultimately kind of discuss the nuts and bolts of how cells utilize the membrane in order to transport materials within themselves, but also between cells as they do their normal day-to-day functioning. So let's dive in and get started. The first thing we need to talk about in order to understand transport is this idea of selectively permeable or selective permeability. Cell membranes, lipid bilayers, are selectively permeable, and what that means is that they are selectively, meaning they allow some things to come in and some things to go out, but they are not fully permeable. So these membranes allow certain materials to pass, and that's really important. Okay, I like to think of the cell membrane as a strainer. In kitchen applications or cooking applications, you often use a strainer to help remove water or excess stock from what it is that you're cooking, and so that's very indicative of how the cell membrane works. The membrane will allow certain materials, certain solutes to diffuse readily through the membrane, but a lot of times the membrane proteins that are associated with the membrane, the channel proteins, the pumps, are the ones, the molecules that are actually kind of actively regulating the materials as they pass. And so we're going to get into that in a little more detail. But some molecules can pass through the lipid bilayer easily. That is diffusion. We can see that here. Okay, no membrane protein is required, just a concentration gradient. So these solute molecules are going to go from a high concentration to a low concentration, and as they are naturally diffusing through the membrane down their natural concentration gradient, they are going to ultimately try to establish equilibrium, and equilibrium would be reached when the concentration of solute on this side of the membrane is equal to the concentration of solute on this side of the membrane. That is the easiest form of cell transport. To get a little more complex, you would employ a channel protein. Notice this is an integral protein. Okay, it's a protein that spans the entire membrane. It would have both polar and nonpolar specific regions that interact with the polar and nonpolar regions of the membrane. But these channel proteins, in a facilitated diffusion application, would utilize the same concentration gradient. Again, we're still going from high to low, which is why it's a passive transport mechanism. These solute molecules would be too large, though, however, to just readily diffuse through the membrane, and so therefore need this hole or this gate in order to pass. And so these molecules, again, would be going from high to low, but would be facilitated or helped along or helped through the membrane through this channel protein that serves as this kind of gate for these materials as they pass. And so some molecules can pass through a channel without energy, but utilize a membrane protein, and so we call that facilitated diffusion. Some molecules have to be pumped through a channel. That is where this image differs from these. Okay, notice that these are high concentrations towards low concentrations. So this membrane or this picture, this diagram of these membrane transport mechanisms would be a passive transport mechanisms. This one would be the active transport mechanism. And so you can see that this ATP is required. This would be energy. We've talked about ATP before. Adenosine triphosphate is the form of energy that cells utilize for a variety of cellular processes. This energy would be employed in order to actively pump solutes against their concentration gradient. Notice that we're looking at these squared solutes, for instance. So if we have 2 solutes here on this side of the membrane and 5 solutes here on this side of the membrane, then obviously this would be the low side for that particular solute. This side of the membrane would be the high side for this particular solute. And you can see that the arrow is showing that these solutes are being pumped from an area of low concentration to an area of high, which is different from these membrane transport mechanisms. So because we are pumping these solutes from low to high, it requires energy that is active transport, and that is going to be readily used or used readily within the cell for a variety of regions, specifically in the action potential of neurons, which we'll get to later in the syllabus. So another form of active transport that isn't pictured here but will be pictured later in the presentation is that very large molecules can be transported via vesicles. That is called bulk transport. Makes sense. We are transporting bulky materials. That is a form of active transport because it too requires energy, but it is not pictured here because while these are active and passive transport mechanisms, these are readily going through the membrane. The bulk transport requires its own vesicle as a transport medium by which to move those bulky materials. So we'll talk about it in a little bit, but I just kind of wanted to highlight what the selectively permeable idea is and why it's important to talk about selective permeability, but also the difference, kind of an intro, the difference between active and passive transport mechanisms. So if we highlight the difference between passive and active transport mechanisms, the first thing that comes to mind is that when you are very passive, you are not requiring energy. A passive anything, specifically passive transport, is a form of transport like these over here that are not employing energy. Notice there are no ATP molecules being utilized. You are going from a high concentration to a low concentration. All of these arrows are pointing from an area of high concentration to an area of low concentration. They are going down their normal diffusion gradient through either the membrane specifically or through a transport protein, channel protein, but most importantly does not require energy. That's very important. It does go from high to low, also very important, which is different than the low to high concentration against the concentration gradient you see in an active transport mechanism. Examples of passive transport that are going to be highlighted are diffusion, osmosis, and facilitated diffusion. Now we've already talked about facilitated diffusion and regular diffusion. Osmosis is a new term that we haven't specifically talked about yet in this presentation. We will. Osmosis is just like facilitated diffusion in that it requires a concentration gradient. It requires a high to low concentration. However, it is just the movement of water and not the solute, which is pictured in these diagrams. Does not require a membrane. However, passive transport can use a membrane. So you can see here that this membrane is intact. The membrane is in the diagram. These solutes require this membrane. The membrane has to be anchored in the membrane, or they require a protein. The proteins have to be anchored in the membrane. However, diffusion can happen away from the membrane. Doesn't have to use a membrane. So it doesn't require a membrane. However, it often does. To contrast passive transport with active transport, this image is showing you active transport. You know it's active transport for two reasons. One, requires ATP. Okay, it requires energy. That energy is usually in the form of adenosine triphosphate, and you can see that the arrow is going, again, like I said in the last slide, against the concentration gradient. Okay, against the concentration gradient from an area of low to an area of high. Again, you can see that this circle solute is in low concentration here. It is in high concentration on this side. So for active transport to work on that solute, it would actually use energy to pump this to that. So you have to kind of pay attention to what solute is actually being moved. This is the solute, for instance, that is being currently moved, and so this is your low, this is your high. It's going to pump it against that concentration gradient. Requires a membrane because it requires a pump or a channel protein that is specifically designed to pump materials against their concentration gradient, utilizing the energy that is released from ATP. Okay, so that is the active transport and passive transport mechanisms. We're going to talk a little bit about each of those mechanisms kind of specifically or by themselves. And the first one up is diffusion. Diffusion is the most simplistic type of membrane transport. Diffusion does not require a protein. You can easily witness or observe diffusion happening by dropping food coloring into water and watching what happens. Why is this showing you or showcasing diffusion? It's simply taking a solute from high concentration and moving it down a concentration gradient to low concentration gradient or low concentration with or without a membrane. Okay, so if a membrane is in place, diffusion can happen readily through the membrane, does not require a protein. If diffusion was happening through a membrane, these solute particles, which are in very high concentration on this side of the membrane and very low concentration on this side of the membrane, will diffuse from high to low through a membrane or without a protein, without energy, as they try to ultimately reach equilibrium. At this point, you can see that the concentration is the same on both sides of the membrane, and so we would say that equilibrium was reached. Okay, that's ultimately the goal of any diffusion or molecule going through diffusion. Here, there isn't a membrane present. However, when you drop the food coloring drops into water, it is obviously going to go from an area of high concentration out to fill the beaker to where it is very low concentration. And again, you can see that it is spreading, and eventually, if enough time happens, the beaker will become uniform color. At that point, there is an equal concentration or an equal distribution of molecules within the solution of that beaker, and equilibrium would be reached in this case as well. Okay, so to kind of go through the next round of passive transport mechanism, we have facilitated diffusion. Facilitated diffusion is just like diffusion in that we are still going from high concentration to low concentration. It is still not using energy, so it is still a passive transport mechanism. But the difference between regular diffusion and facilitated diffusion is that facilitated diffusion is helped or facilitated by a specific protein. And these proteins come in two forms. Protein channels, or channels that span, they're integral proteins but span the entire membrane just as an open hole through the membrane, kind of a pore through the membrane. Or carrier proteins, which are much like what they say, they carry specific solutes from an area of high concentration to an area of low concentration, but they're actually kind of actively moving or opening, whether they're open to the external to internal or internal to external, but they're going to carry molecules as they move or change conformation in the pumping of those materials. But again, they are carrying the molecules, they are not pumping. I shouldn't say pump. Pump is more indicative of an active transport mechanism. But you can see in this image there are two specific types of these intrinsic proteins as we move materials from a high to low concentration. Okay, but the key is we are moving materials down a concentration gradient without energy from an area of high to an area of low concentration. Osmosis is, much like I said in the beginning, the passive transport of water specifically, and so you will often see a specific substrate or a specific solute associated with the water because we often don't have pure water. However, osmosis is the specific type of diffusion involving only the movement of water across a selectively permeable membrane, and often the membrane isn't permeable to the larger solute molecules. It is just permeable to water, and so the water is going to move with its concentration gradient or down its concentration gradient from an area of high concentration of water, to an area where the water is less concentrated, okay, so low concentration. Notice that the arrows, there are more arrows moving from left to right in this image than there are right to left. And we'll talk a little bit more about that in a minute, but the water is going to move down a concentration gradient, so without energy. It is not pumped. It is not really going through a channel protein that is using energy to pump the molecules from an area of low to high. But it is going to move down the concentration gradient from an area of low solute, which is high water, to an area of high solute or low water. Again, we're going to key in on the movement of water. So we're going to go from where water is more highly concentrated to where water is more lower, okay, or where it's lower concentrated. Water always moves from a hypotonic to a hypertonic solution. Again, we'll talk about what those terms mean. But once isotonicity is reached, basically there are three terms: hypotonic, hypertonic, and isotonic. Once isotonicity is reached, water molecules move back and forth in both directions equally. Water does not ever stop moving, which is why even when there is a concentration gradient established, we still see arrows pointing in both directions. However, the majority of the movement, okay, or the net movement, the majority of the movement is moving from an area of high to an area of low, and again, it'll make more sense in a minute. So if we highlight these images, water molecules, which are indicated by these little blue dots, are going to be moving from where they are more highly concentrated to where it is lower concentrated. And so you will see that the majority of these arrows are pointing left to right. These water molecules want to balance out the concentration on both sides. They want to reach isotonicity. And so these water molecules are going to just naturally diffuse from where they are high concentrated to where they are low concentrated. As the water molecules move in this direction, more of these water molecules are going to come this way and are going to therefore cause this water level to rise, therefore diluting out the solute that is in this side in order to try to equal out both sides. Okay, so if this is 100% water and this is 50% water, it wants to increase the number of water molecules on this side so that it drops the solute concentration from 50% to 60, 70, 80, 90%, right? Obviously, water is going to keep moving. If it was able to reach isotonicity, which means the two sides of this membrane would be isotonic, meaning they contain the same concentration of solutes, then the arrows would be equal. There would be equal arrows pointing left to right as there are arrows pointing right to left. At that point, net movement of water is 0, but every time you had a water molecule move to the right, you would have a water molecule move to the left. So the movement of those water molecules don't stop, but the movement would be equal once isotonicity is reached. Okay? If we look at diffusion within a different image, and we utilize this J tube, which has this unpermeable membrane in this particular diagram, two things are going to happen. Depending on the permeability of the membrane. Unpermeable means that it is not going to be permeable by both, and so the water on this side is going to stay on this side, and the water on this side is going to stay on this side. And notice that we can actually establish two different concentrations of solutes, in this case salt water, right? So the solutes in this case are salt, so we can keep a higher salt concentration on this side than this side. The movement of water doesn't happen because the membrane is unpermeable. However, when we create a permeability, or if we open up a permeability with the membrane, then we start to see something happen. If the membrane is permeable to everything, water will not only move from an area of high water to low, water will move in this direction, but the solute is permeable as well, or the membrane is permeable to the solute, and so while the water is moving this way, the salt is moving this way, causing both to equal out, and so we reach equilibrium. Okay? Everything diffuses from high to low. So because this side of the membrane had a higher water concentration and a lower solute concentration, the water wants to go from where it was more highly concentrated to where it is lower concentrated. Water moves this way. Salt was more highly concentrated here than it was here, and so the salt, while the water is moving from left to right, the salt will be moving from right to left. So salt, the net movement of salt moves in this direction, the net movement of water moves in this direction until ultimately equilibrium is reached. That is in a purely permeable membrane, which often isn't the case in real kind of biological contexts. Usually what is more realistic is the semi-permeability of a membrane, and what that means is that the water is permeable, okay, or the membrane is permeable to water, but the membrane is not permeable to the solute. When the solute is not permeable, the solute molecules are not going to be able to pass and therefore have to remain on the original side. If you were to count up all of these red dots, it would be identical in this image to this image. If you were to count up the red dots, they would be identical in this image as they are in this image. The only difference, and the reason why the concentrations look different, is because the movement of water happened while the solvent was remains stationary. right? So the reason we have a displacement of water is that water was more highly concentrated on this side of the membrane than it was on this side, and so water wants to move in that direction, causing the water on this side to drop, causing the water on this side to rise, and by increasing the volume of water on this side, we have caused the solute to become more dilute, and by dropping the water on this side, we have caused this solute to become more concentrated, therefore equaling out the concentrations on both sides, still reaching equilibrium. Okay. Now, if we look at it on, or in terms of this image, it's doing exactly the same thing, only here the solute is sugar concentration. We have a dilute sugar concentration, we have a concentrated sugar solution, and we have a membrane that runs right down through the middle. If the membrane is, again, selectively permeable to just water and not solute, the water is going to move from where it is more highly concentrated here to where it is more lower concentrated on this side, causing the net movement of water to move in this direction, causing the volume to decrease on the right side, causing the volume to increase on the left side, and therefore trying to reach equilibrium through the more concentrating the solutes on this side and the dilution of the solutes on that side. Why is osmotic control vital? Many of the medical procedures we do today are the direct result of osmotic control. And so, again, it is vital. There are a lot of life and death situations that result from the simple passive transport mechanism of osmosis. And so some of the medical procedures that require osmotic control to prevent damage to cells and tissues would be through IVs. If you're dehydrated or you go into the hospital and require fluids, they are often going to provide you IV fluids, saline, that are often isotonic or slightly hypotonic, meaning we want to semi-force water into your blood cells, but we don't want to do it in a way that causes cell damage or tissue damage. Okay, if you just put pure water into somebody's bloodstream, it's too hypertonic, or the solution is too hypertonic to the water, or I should say the solution is hypotonic, right? And it's going to cause all of that water, which is pure and free of solutes, to become readily absorbed by the blood cells, and they therefore swell and can burst, which obviously is bad. You don't want your blood cells bursting when exposed to IV fluids. Wound care. Washing a wound is often done with an isotonic saline solution. Again, you want it to be isotonic because you want the cells to remain normal size. You don't want to subject them to a hypotonic or a hypertonic solution because you don't want them to shrivel or swell. Eye, nose drops, again, you want those to be isotonic. And then organ transport, when organs are removed by a donor or from a donor and transported to the recipient, they are often going to be housed within an isotonic solution, again, to help retain normal cell hydration levels, but normal cell size functioning, right? We don't, again, want to produce a swelled or shriveled cell. Now, how do these images, or what do these images really mean? Well, when you subject a cell, let's say, for instance, okay, a red blood cell, to a hypertonic solution, what does that term mean? Hypertonic. Hypertonic is exactly what it sounds like. Hyper is an increased solute and therefore decreased water. Hypotonic is the opposite. So it's a decreased solute and an increased water. And then isotonic, obviously, you have the same water and same solute concentration. So I'm just going to focus on hypertonic, hypotonic. So let's say the cell is 90% water, just for conversation's sake, and we'll say it's 10% salt. Okay, I know the cells have other things, but let's just say there's a 90% water and there's a 10% salt solution within the cell, or based on the makeup of that cell. If we subject this same cell to a hypertonic solution that has higher solute concentration and less water, this hypertonic solution could be, let's say, 20% salt and a 80% water. Well, if we subject this cell to this solution, the solute concentration is higher in the solution than the water, therefore making the water concentration in the solution lower than in the cell, therefore causing water to go from high to low. 90% water is higher than 80% water, and so water is going to leave the cell because it naturally wants to diffuse from an area of high to an area of low, causing the cell to shrivel because it is losing its water, which is why up here in this image, when you subject a cell to a hypertonic solution, you cause the cells to shrivel because the water has left, or the water is leaving them, and you essentially get the kind of raisin effect from the grape. To contrast that, when you subject the cell to a hypotonic solution, you get the opposite effect. So solute is decreased from what it was in the cell, and the water was increased from what it was in the cell. And so the water, let's say, is 95%, leaving the solute concentration to be 5%. Well, solute is less in the solution now than it was in the cell, and so the water therefore is higher in the solution than it was in the cell, and so water naturally wants to diffuse from where it is high to where it is low, 95 to 90, and so in a hypotonic solution, water is going to flood in, causing the cell to expand or swell, and eventually, depending on how much it takes in, can burst and or lyse, and therefore cause the contents to be expelled, and those cells obviously are damaged, and that can lead to not only cell damage but tissue damage, again, which is why osmotic control is vital. So hypertonic, hypotonic, very important. Isotonic is the other term. That is, though, when you have the same concentration as the solution the cells are in, therefore really just not causing a net or an excess of water intake or an excess of water loss, causing the cell to take in as much as it's giving away, leaving the cells to remain normal, functioning, normal structure. Okay, so the last thing we need to talk about is active transport specifically. And so active transport is very different from passive transport. It still requires a membrane. It still requires a protein pump or a protein channel, a carrier protein that is embedded in the membrane. It is going to move the materials against the concentration gradient from an area of low to high. The reason it requires energy is because it is moving the solutes against their concentration gradient. Everything naturally without energy wants to diffuse from an area of high to low. When you start deviating from the norm, you obviously have to go against the grain, so to speak, and in order to go against the concentration gradient, we have to employ energy, and often the energy source when we talk about active transport is in the form of ATP. So how does this active transport work? One of the things I want to talk about specific to active transport is this sodium-potassium pump, because throughout biology and throughout the IB syllabus, you're going to see this sodium-potassium pump utilized in a variety of contexts. And if you have a solid understanding of how the sodium-potassium pump works in an active transport setting, then I think you're going to be able to apply it to any of the other situations that require the sodium-potassium pump, and we're not going to have to kind of further explain it then. So, active transport obviously requires energy. The sodium-potassium pump is an active transport mechanism, and the one kind of application of this active transport that I want to talk about in detail. So there are about 6 steps that we need to talk about, and so step 1, just to break this process down, is 3 sodium ions. Notice we have sodium ions that are blue, and we have potassium ions that are kind of this purple pinkish color. 3 sodium ions are going to enter this channel protein or this pump protein from inside of the cell cytoplasm. Again, cytoplasm is the material that is inside of a cell. Extracellular space is the space outside of a cell. So the sodium ions, 3 of them, and 3 is important, okay? 3 sodium ions are going to enter this protein pump from inside of the cell and attach themselves to the binding site of the protein. Notice these proteins have 2 potassium binding sites and 3 sodium binding sites. The 3 sodium ions are going to bind to the sodium binding sites of the channel protein or the pump protein and are therefore going to cause this protein to change shape. Once the shape is changed, ATP breaks apart and transfers a phosphate to the actual protein. The phosphate that breaks off of this ATP, along with the energy burst that happens as the phosphate breaks or separates itself from the ATP, is going to help that protein change shape, and it causes the protein to open to the opposite side. So in this case, it's going to open from inside to outside, and when it opens to the outside, it's going to release the 3 sodium ions, 1, 2, and 3. It's going to release those 3 sodium ions to the outside. Notice the phosphate is still attached, keeping this conformation open to the outside. Once those 3 sodium ions are released to the outside, because it is still open to the outside, the channel protein is still open to the outside, 2 potassium ions are going to enter from outside and attach to their binding sites, causing the phosphate to jump off or be removed. At that point, when the phosphate removes itself, the 2 potassium ions are going to then leave because this protein changes shape. They're going to be released to the inside of the cell, causing the pump to return to the original conformation and therefore opened up to the inside, causing these sodium ions to then enter the channel protein or the pump protein, and therefore starting this whole process over. Why is this an active transport process? Again, because this side of the membrane is low sodium, right? We have these sodium ions. There are a lot more sodium ions, blue sodium ions, outside of the cell than there is inside. So we are actively pumping this from low to high with energy. Again, the potassium ions, it is low on this side and high on this side. Okay, so again, we're moving these from low to high. We're moving these low to high. Both of them are being pumped low to high with energy. It is a form of active transport. This is the way the sodium-potassium pump works. It's really important for biology as a whole, especially the IB syllabus. Okay, at the very end, 2 potassium ions leave the protein pump and are released to the cytoplasm. Again, they were released back to the high side. We're pumping them against the concentration gradient, and that allows 3 new sodium ions to enter, starting this whole process over. So those are the 6 steps. I would know those 6 steps. I'd be able to talk through a diagram, or at least be able to write these 6 steps on an extended response question. Really, really important to understand active transport through the lens of the sodium-potassium pump. The very last thing we're going to talk about in this entire membrane transport video is the idea of bulk transport. Bulk transport is exactly what it sounds like. We are transporting bulky materials through the cell. That can be into the cell and it can be out of the cell. Moving bulk materials into the cell is called endocytosis. Moving bulk materials out of the cell is called exocytosis. Okay, you can see that in these diagrams here. Endocytosis is the invagination or the pinching in of materials from outside in. If these are materials we want to bring into the cell, these materials are going to come to the membrane. They're going to push into the membrane, causing an invagination of the materials with or using the fluidity of the membrane to ultimately form a vesicle or vacuole, and therefore now we have materials completely surrounded by a membrane. This membrane was once part of the plasma membrane or the cell membrane, but now we have a way to kind of wall off or compartmentalize these materials that we just brought into the cell. We can then use this vesicle to transport those materials to where they're needed, anywhere in the cell. Exocytosis is the exact same thing, only it's taking materials that were produced by the cell, moving them up to the membrane, and then basically fusing the vesicle to the membrane, opening up a hole in the membrane, and then releasing those materials out of the cell because they ultimately are going to be used out of the cell, or maybe are byproducts of certain enzymatic reactions. but it's all about secretion of contents. So we're either going to bring in bulky materials by the invagination of the membrane to form a vesicle, or we're going to take a vesicle with contents produced by the cell and are therefore needed to be secreted to the membrane and secrete or release them out of the membrane. Both cases require the fluidity of the membrane because the membrane has to be able to move. The membrane has to be able to accommodate the vesicle that is either coming in or leaving the cell. Within the idea of endocytosis, there are three types of endocytosis that I want you to know about. Phagocytosis is cell eating. Okay, so I want you to think about cell eating, which is the invagination of kind of solid particles. Notice solid particles. It technically isn't really eating in the context we think of eating, but if you think about cell eating, we eat solid food. We don't drink solid food, right? We eat solid food. Solid particles are going to be brought in through the process of endocytosis, specifically kind of with a sub-process of phagocytosis. Fluids, okay, extracellular fluids, fluids that contain solutes or small particles are going to be brought in through endocytosis, but specifically with the process of penocytosis. Penocytosis I would consider to be cell drinking because it is fluid and not solid. Cell drinking would be penocytosis, cell eating would be phagocytosis, and then there's a different form of endocytosis that's all about receptors and receiving signals. These receptor proteins, which are on the surface of our cell membrane, will receive these signals. Once they receive and bind to these signals, they will invaginate or create this specific vesicle that contains the receptors that are bound to the receiving molecules, and therefore this is a very special purpose, or has a very special purpose, very special functioning that these did not have. Okay, so this is bringing in food per se, solid particles, this is bringing in fluid, drinking per se, and then this is bringing in a specific set of receptor-mediated signals that we're then going to utilize within the cell to cause some form of functioning to happen. Okay, so receptor-mediated endocytosis, penocytosis, phagocytosis are all three forms of endocytosis, which is kind of the generic form of bringing in materials through this invagination of the membrane. Have to be fluid. You require the membrane to be fluid. It's one of the reasons why the membrane is fluid in the first place. But that's it. That's where we're going to end it. Bring your questions to class. See ya.